Hello, BNG426 526 class. Hopefully you had a nice long weekend this weekend. I apologize for getting this out to you late. I know our flipped classroom is scheduled for tomorrow. I really had planned to get it out earlier. Unfortunately, my son very kindly gave me the flu this weekend. So really this is the first opportunity I've had to actually record a lecture and not spiral into a coughing fit. So here we go. This flipped classroom is going to be about metabolic networks. We've been talking about metabolic networks and their makeup. So we can take an interest in a gene or an enzyme, a product most likely, or a pathway to make that product. And we can study it, we can improve on it. If it's a product, we can make more of it. We can subject to the organism of choice, whatever our microbial chassis is, to perturbations in order to, again, quote unquote, improve on what we're interested in. Perturbations affect more than just the gene or pathway of interest, though. They sometimes even have unintended consequences. Metabolic networks can be examined, and they can tell us about what we're doing, how that affects other pathways, or even the cell fitness in general. So it can give us clues about energetics, about maybe how this might affect cell division, depending on where we're playing, maintenance, cell maintenance, storage of reducing power, which is a big deal if we're talking about things like ethanol or polyhydroxybutyrate or something like that, and stress responses. And we can study these things beyond just the pathway level, which is what we're doing. We're studying them in the network level. So you look at a network, and you look at it you step back and you squint your eyes and look at it, and it just looks a bit unwieldy. Like I don't really know what's going on here. But in this example, even though it's a lot of data, it's a lot of things to take in, we can see how an oral bacterium will respond to you being sick. So there's a lot of things involved, and there's a lot of things that are happening. And you can see the pathways in yellow are affected the most when it comes to an oral bacterium responding to how you are when you're sick. But how do we get there? How do we get to that? To having a giant map that has either different color arrows or different color lines and dots, or heavier lines or what have you, that indicate that a perturbation has happened and that this perturbation affects this, that, and the other pathway in our organism of choice. How do we get to that? How does a network first come into being as a thing we study? It's simple enough to say that the entire cellular metabolism is a network that is a thing we study. It's not entirely true, because I'm sure we don't care about the lot of it. A lot of the decisions we make in Saccharomyces cerevisiae to produce ethanol don't affect other metabolic pathways. Some do, but some don't. But it's, how do we establish a network, and kind of make it tangible enough so that this is what we care about and it affects our goals, our short-term and long-term goals for the production organism or what have you. And so then again the question is, in terms of experiments, what do we need to get enough data to begin constructing a network? Is it enough to just have a genome sequence? DNA sequences, are they enough? Do we need transcriptome data? Do we need metabolome data? Things like that. Do we need proteome data? So every network does start with a genome. You do need a little more than that. It's hard to construct a network if you don't know what genes your organism has. So it's a pretty good idea to have a genome. Genome data is relatively easy to get these days. It's especially easy in bacteria. There was a time, back probably 20 years ago, when genome data was a pretty big thing. regardless, and it took a collaboration of 5 to 10 different institutes across the world to sequence the entire genome of E. coli. And this was a huge thing. But nowadays, all I have to do is make a prep of a random organism that I find in soil or in my mouth or something, and send it off, and maybe a month later I get enough sequence to work with that I can put it on NCBI and publish a little genome announcement paper and pay $300, and there we go, another genome. And it takes me a month or two, whereas it took 5 institutes maybe 5 to 10 years back in the 90s. But anyway, regressions aside here, networks start with the genome. It's helpful to know what genes your organism has. But you need additional reinforcing data to establish your network. Things like gene expression, transcriptomics, that can come in the form of microarray analysis, RNA-seq analysis, proteomic analysis, metabolomics, even comparative genomics. We can compare different strains to each other and different species of the same genus to each other and see what one has that the other doesn't have, and vice versa. And so we can test these networks and see how they stack up. So we can look at a bunch of different ways to do this, and we can try to get some refinement of our metabolic network. So we can, with methods A, B, and F, we have our, we can do knowledge gap filling. So if we have a metabolite that's being made, we don't know, where it fits in. It's helpful to test our networks in order to be able to, put that piece in the puzzle. Contextualization, it could be that stray metabolite is only made under certain conditions, and it's helpful to know that. So, that can help us refine the network even further. And then there's those species comparisons. Those can help as well to refine our network and make it a sturdy network that we can use going forward in our design of our production organisms and things like that. Okay, so we can look at this extremely abstractly, which is what we're doing here. So we can look at, say, hey, we have some metabolites. Let's make a network out of them. How do they fit in? Say we have a tiny organism that makes 9 metabolites. It's a, as far as we know, it's a random assortment of metabolites. We just know that the organism makes them in some degree. Some are our parameters or our substrates and products. Some are just our variables, our intermediates. So we know we care about some of them, maybe all of them, but how do they relate to each other? And so when we're talking about a network, just like you and your Facebook friends, there are degrees of separation there. How many degrees of separation are there between the different metabolites? And we talked about clustering coefficients and things like that. But here we just simply want to put the pieces of the puzzle together and see how they fit. So we can address this in a variety of ways. There's a few different techniques we can do to begin to assess this. So first of all, we can start by saying, well, we're going to be interested in this pot of 9 metabolites for a reason. So we're interested in metabolites number 2 and number 9. And I will tell you right now that number 9 is our substrate and number 2 is our product. So that kind of puts some context here. So we've got these 7 other metabolites, but we now know that number 2 and number 9 are important because we've singled them out, number 9 being our substrate, number 2 being our product. And there's a pathway in there somewhere to get number 9 to number 2. So likely some of the other metabolites will be closely involved in doing this, in making number 2 from number 9. Maybe all of them will. We'll have to find out. There's ways to figure that out. So initially we could do some stoichiometry. It's pretty easy to know what the molecule of number 9 looks like, just like it's easy to know what the molecule number 2 looks like. And it could be, in this case we'll say that it is, that for every mole of 9, 2 moles of 2 are produced. So essentially we're reading that as 2,2 on the grid. And this is important if we're examining the control of the flux. We need to know that stoichiometry, as you know from when we did flux control. So first we look at genomics. We can examine the genome of the organism of interest. The gene annotations will, to some degree, tell us that certain genes might have certain names. In this case, annotations can give us an idea of what the pathway looks like. So it could be that we see here number 9 to number 8. This could be, this gene that makes this enzyme could be a 9-ase that takes 9 and makes 8. And 8 to 5 here, that could be an 8-ase, let's just say. And that makes number 5. And then eventually this enzyme, this hypothetical enzyme at this point, makes 2 moles of 2 out of 1 mole of 5. Let's just say. So this is giving us an idea of what the pathway looks like. Are we done? Not by a long shot. But we have a little more of an idea of where we're heading. So I mentioned gene annotation. What does that even mean? Annotation of genes is not an exact science. Anyone can annotate genes. It's often just about educated guesses. Oftentimes, you dump these things into a computer program, just like I recently published a genome sequence. I dumped a bunch of genome data into NCBI. They annotated it for me. They made a bunch of educated guesses because there are some conserved regions that look like they might belong to certain gene groups, their gene products might belong to certain classes of enzymes, something like that. But the fact is, you don't know for sure. So annotation of genes could just be like a good mystery or a crime scene investigation. The more evidence you have, the more likely you will be to get the prediction correct. So if we have more than just, well, this is what the gene looks like, if we have, some proteomic data, and hey, it turns out we've actually purified the enzyme and run some enzymatic assays on it, well, we're getting there. We're in the right ballpark to say that, hey, maybe this is what this enzyme indeed is, and that our prediction is more likely to be correct. Or we could knock it out and see how that affects the pathway. So gene annotations, in the absence of physiological or biochemical data, either knocking out the gene or doing enzyme assays or proteomics or something, these gene annotations, in the absence of any of that, are predictions. They're not really binding. Although, in some cases there are significant similarities and the predictions are pretty good. But still, they're predictions nonetheless. So other things we might want to ask about our pathway here are, is any part of the pathway reversible? Are there carbon sinks to consider? Are there other ways to make our product, our number 2? How does the key pathway of interest fit in with this cellular metabolism? So biochemical data can tell us a lot about whether a reaction is reversible or not. The only caveat is, and we've talked about this already, in vitro enzymology is not the same as an in vivo functioning pathway. So we can isolate all these enzymes and show that they're reversible, but that does not necessarily mean that they're going to act the same way in the cell. So we can show that this 8 to 5 enzyme or 5 to 8 enzyme is indeed reversible in a tube. Same with this 8 to 9 enzyme is reversible in a tube. It may well be that's what it is in the cell, but we can't be 100% sure. But it does give us a little added data in our quest to give more granularity to the pathway here, to the network. So ideally, biochemical data and metabolic physiological data should confirm directionality of a reaction. And there are ways we could do that as well. We could confirm bidirectionality. Potentially we could feed 5 and see if we can make 9. Who knows? Then that may not work, but who knows? There are things we could try anyway. But for our example, let's just say biochemical and physiological data confirm that 9,8 and 8,5 are reversible. So we put double-headed arrows on them. Whereas 5 to 2 to the 5 to 2 twos is not reversible. It's irreversible. So that stays with the single-headed arrow. In the end, transcriptomic data, high expression of genes under certain conditions, can suggest or confirm roles in the network. So if we look at transcriptomic data when we feed the cells product 9 here, or substrate 9 I should say, we see that there's a gene that's highly expressed that takes metabolite 7 to metabolite 5. So if under conditions favorable for the production of metabolite 2 or favorable for the utilization of metabolite 9, the enzyme responsible for making number 5 from number 7 is upregulated, then maybe it should be a part of the network because you're feeding into 5, which eventually feeds into our product, the product we care about, metabolite number 2. And sometimes there's just no substitute for experimentation. You examine some growth and metabolomic data, and you find that number 1 is also made under conditions favorable for the production of number 2. On its own, that doesn't mean a whole lot. So, some radiolabeled substrate experiments can show that at least a fraction of number 1 is made using number 5 as a precursor. So, sorry to those of you that thought we were getting a bonus amount of number 5 to feed to number 2 because of this 7 to 5 link. Of course, we're not really saying where 7's coming from at this point. But some of the number 5 is going to make this, we'll call it a byproduct, this number 1. So we can look at different types of physiological data. So we can look at radiolabeled substrate and see where the label goes. If it goes into number 1, then we know it's going into number 1 through the pathway needed to make number 5. So if given it labeled 5, then we've shown that okay, well we make labeled 2, but we also make a little labeled number 4, and then we're ending up with some labeled 1 here. So we're learning that these metabolites are linked in our little network here. And then we can compare our little organism to other organisms of this size. We can see how other closely related organisms do their business and how that can help us uncover how our organism of choice operates. And if strain B of an organism has a recycle pathway that feeds off of and into our pathway of choice, then perhaps our organism can do that too. And if those metabolites happen to be 6 and 3, well then we've seen how 6 and 3 can fit into this whole thing. And so in our case, the metabolomic data has shown this. That we have this little recycle pathway, which may look like a futile cycle, but maybe it's not. Maybe 3 is being used for something, we don't know. So in other words, we've used a bunch of different types of data that all are trying to get at basically the same thing. We've used them all to get at what our network is entailing. What is involved in our network. It's more than just taking 9 and converting it to 2. We've got an extra feed here with 5. We're siphoning some off, but then it's coming back. We don't know how much we're losing out here. And, we're making product, but we're also making byproduct too. So there are many ways to unearth a comprehensive metabolic network. Now, I'm not asking you to know what I'm not asking you to know the different metabolomic data gathering experiments can be done. I'm not asking you to know what radiolabeled substrate is all about. Things like that. You don't need to know the molecular biology or the biochemistry of that stuff. But just know that there are multiple ways which we can do experiments that can give us an idea of what it is we're looking for, which is to find out what the players are in our network and more or less what their roles are. And so different experiments can show metabolites with a relationship to our pathway of choice. Now, when we started, our pathway of choice just involved 4 metabolites, but we soon found that all 9 metabolites were involved somehow in this. Is this the end of the story? Probably not. We can use genomic and gene annotation data to set up our network of choice, but then we can refine it. We can further refine it by a bunch of different types of experiments that we can do, or data that we can gather. You could probably go into the literature and gather a bunch of different data from organisms like E. coli, organisms like Saccharomyces cerevisiae, and figure out different types of networks there. So many high throughput experiments can help us refine and add to our pathway. Okay, I'm about to lose it. So let me get right to the to-do. So each team is a group of collaborators working on the same network from different directions. Thus, and this is the important part, if I can get it out, each member of the team will get a different paper, A to D. Genomic data will tell you the metabolic pathway from substrate to product. Each individual will report findings to the team, and each of you, A, B, C, and D, will have different findings to report. Different types of experiments will yield different types of data. As you report your findings, the network will take shape. So all of you in the team will draw out your network based on a simple grid like we had in our small example here. It'll be a little bit bigger, but the idea is the same. And you'll use those data to put together a pathway. And then you will talk about how many individual pathways you can find within that one network that you've made. So, thus, it'll be important tomorrow to get in your teams, and then I will give each member of the team a different paper, a different assignment paper, which will be numbered 7A, 7B, 7C, 7D. And then from that, you will all collaborate and put together your network. And the deliverable at the end of the class time will be each individual member giving me the answers to the questions on the assignment and the map of the metabolic network that you've made based on those data. So in that case, before I spiral off into another coughing fit, I will talk to everyone tomorrow and not cough on everyone tomorrow.